And we would like to ask you, if you have not muted, to please do so. We are pleased to have you join us today, and the title of today's presentation is Estuarine Acidification, a conceptual discussion with examples. This series is hosted by Southeast Ocean and Coastal Acidification Network. In short, the series lays a foundation for the state of ocean acidification science in the Southeast region. These webinars are intended to create a dialogue among scientists to identify what is known, what isn't known, and what research in other regions of the United States can be applied to better understand ocean acidification and its impacts in the southeastern U.S. region. With awareness of and access to the research and its applications and uses, webinar participants will be able to collaborate to better understand and adapt to ocean acidification moving forward. As I mentioned, I'm Paula Keener. I'm Director of Education for NOAA's Ocean Acidification and Research Program and its Office of Ocean Exploration and Research, and a member of the SOCAN Steering Committee, and I'll be facilitating today's session. We also have Jen Bennett-Mintz, and she's Education and Outreach Coordinator for NOAA's Ocean Acidification Program online, and Jen will be helping to answer questions from the attendees. During the presentation, you as attendees will be in listen-only mode. You're welcome to type questions related to technical issues or questions for the presenter into the questions box in the control panel on the right-hand side of your screen. We'll be monitoring incoming questions during the presentation, and we will respond to them or pose them to our speaker after the presentation. We are also recording this session, and the video recording and the PDF of the presentation will be available on the SOCAN website. We are pleased to welcome Wei-Jun Cai today for the presentation. Wei-Jun is a professor at the University of Delaware School of Marine Science and Policy in Newark, Delaware. Wei-Jun received his PhD in oceanography from Scripps Institution of Oceanography. And Wei-Jun has worked on marine carbon cycling for 20 years. His research areas include calcium carbonate dissolution and sediment diagenics in the deep sea, using microelectrodes, air-sea exchange of carbon dioxide, and carbon cycling in coastal oceans. Most recently, Wei-Jun's research has focused on the responses of coastal ocean carbon cycles and ecosystems to a changing terrestrial export of carbon and nutrients, as well as bottom water acidification in estuaries and coastal oceans. Wei-Jun has over 101 peer-reviewed scientific publications. He's received over 31 research grants. He advises numerous graduate students and postdocs, and he has also received numerous honors and awards. Wei-Jun, I'm going to turn the presentation over to you at this time. Welcome.
Introduction. Good afternoon. Can you show me my screen? Hello? Can you hear me? I guess so. Yeah, and thank you to Paula for the nice introduction. While Paula was talking, I noticed I should have put a background on this first slides from, say, Sapporo Island, but for now, let's pretend this is right outside the Altamaha River. You have the plume and the estuarine. So I just want to mention, before I moved to University of Delaware, I was a faculty for 18 years in the University of Georgia, and so that's where I started the carbonate the CO2 system in the southeast. So this was a really happy opportunity, and I'm grateful to this to talk about ocean acidification in estuarine and coastal waters. And so I have some result from the southeast. I can talk about that, but mostly I want to bring a view from different estuarine to see what kind of different acidification we may expect. I understand that last week our first talk, Rick Warnikoff gave a first talk. I promised to go over his webinar over the weekend. I haven't really finished, but I saw the first few. And so he said he will provide a far-side view, and I guess under that context, I'll do a more near-side, I guess. Well, no, I will have both, I guess. Anyway, I'll do a brief introduction, and then I'll start with examples of acidification and low oxygen events in coastal ocean. I was reminded that the audience, most have science background, although not everyone is doing the OA study right now, so I should start with something easy to understand and have. I don't know if I can accomplish that. I'll try. So I'll start with real example, and then I'll go into something more theoretical, try to discuss from first principles why we would see this and that. What do we expect? Changes in the ocean acidification, or how different estuarine will respond to ocean acidification and other biogeochemical process. And I'll have a summary. If we have a few minutes left, I just want to show a few slides of my recent work in the a little bit north of the SAB, so in the Delaware and Chesapeake Bay. All right, so what is really ocean acidification in coastal waters? There seems not a lot of debate about what is ocean acidification in the open ocean, but in coastal ocean it's getting more complicated, I guess, or less defined. Well, first, I want to satisfy open ocean people to say that ocean acidification can still define as what they define, global ocean uptake of atmospheric CO2 as a result of this anthropogenic CO2 increase in the ocean. So that's already lead to delta pH decrease. I think you are seeing two screens. Let me just duplicate the slideshow this way. Okay, so here I would take this more narrow definition of ocean acidification first as a response to anthropogenic CO2 increase in the ocean in the atmosphere. Let me see if we could use this laser point. That was good. So anyway, we have this source water being acidified with higher DIC to alkalinity ratio. Basically, when you uptake CO2, you have higher DIC, but that doesn't change the alkalinity. So this source water, open ocean water, has this higher ratio than pre-industry, and that water would advect onshore and mix with low salinity waters. Now, we could also have local anthropogenic CO2 uptake in the estuarine zone or coastal water as well, but mostly it's probably from, definitely from open ocean. And this water is further acidified by respiration, that is decomposition of organic matter falling from productive surface water, in this case, or actually it could be from terrestrial or productive salt marsh. So we already know there is this acidification of source water from anthropogenic CO2 and this respiration, that is decomposition of organic matter from productive surface water, like in the Gulf of Mexico, Mississippi River plume case, or in maybe outside near Secoora Island, Altamaha River, we could have salt marsh organic matter coming out and get decomposed to produce a lot of CO2, and there are also other microbial process could generate acid. The real question, or less known question, how do they interact? Okay, and I want to emphasize that this interaction, is changing with time as the anthropogenic CO2 increase. Actually, this overlap part will increase. So let me start with a few examples around the North American continent from west coast to first go north to the Bering Sea and come back to the east coast starting with the Estuary and the Gulf of St. Lawrence and down to the mid-Atlantic Bight and to the Gulf of Mexico. Of course, I wouldn't forget South Atlantic Bight. I want to apologize, this America centralized view, if there's anyone from Europe or Asia listening to this talk. All right, so this is in the west coast, the dominant process, as you read from Dick Feely's paper, 2008 paper in Science, is really by this upwelling that brings low pH and low oxygen and low aragonite saturation water on shore and expose the ecosystem there under low pH. So this upwelling water could further actually gets into estuary, like here into the wonderful car and this estuary near Seattle. And you see very low oxygen and very low pH and low aragonite saturation. That is mostly due to this upwelling and superimposed on that upwelling signal is a local respiration. And that this a little kind of stagnation of the water, so allow this low pH to be developed further. And if we move north to the Bering Sea, in the summer case, this is a paper by Jeremy Matthews, and you can see that the low aragonite saturation state is largely driven by this very high production in the surface when organic matter settles in bottom and decompose, generate CO2 and low pH and lead to low pH and low saturation state. And that is also have impact in this seasonal ice melt, and that's also dramatize this low pH events by largely to promote this biological production in the surface. So if we come to the east coast, this first example I show is from St. Lawrence Estuary Gulf, and again for the same reason, this water stratification lead to low oxygen and low pH events in the subsurface and low aragonite saturation state. And what's most interesting from their example is they have some data not only back to 1980s but also to 1935 before pH, the MBS scale was even established, assuming we know the pH relatively well then. So you see this pH decrease with time, a long record. All right, let's move to the South Atlantic Bight first, and I would only mention there are a few past publications from my own group. So this is my former student Wei-Jun Cai's publication to show this saturation state in the U.S. East Coast, mainly in the South Atlantic Bight, and in comparison with the West Coast. And this is a paper, more recent paper by Alec Wang. He actually published this. This paper is about us. This called GOMAC-1 cruise that research led by Rick Warnikoff, which surveys the entire East Coast and Gulf of Mexico and East Coast to show this pH and carbonate saturation state. And earlier, my own research on the inside the estuaries mostly, that was kind of a little before the acidification time, and I actually was one of the first, I guess, point out in the southeast, these river and estuary with rich in organic matter, humic substance. That organic matter can contribute to alkalinity and to the pH, and even control the pH in these estuaries, and we'll show that result very soon. So here are some recent survey. We had 6 cruises in 2014. We haven't really done a good data analysis yet. I just show some preliminary result here. And you can see, as we know before, in offshore, pH are generally high, but in the nearshore part, in the estuarine, inside estuarine, you'd see very low pH to as low as, here we actually show only like 7.5, but in specific measurement, some area it could be very low, like inside here. And almost every season you see low pH in estuarine. And I just added this slide 10 minutes before my talk. This is from my 1998 GCA paper. And in that paper, we show very low pH in the Satilla River estuarine and Altamaha River. So you can have pH as low as 5.5 to 7. These very low pH, and as I showed in that paper, was really dominated by export of the humic substance from swamps and from salt marsh. And there, mechanistically, you have this alkalinity. Surprisingly, 50% of alkalinity is really organic alkalinity, and that organic alkalinity maximizes in the estuarine, low salinity zone, and as salinity increases, and the system gradually shift from humic substance controlled pH to carbonated system to the offshore water. And when I published that paper, of course, we don't know anything about, we didn't really talk about ocean acidification. And so for 10 years, nobody really cited my paper, and until a more recent time, and I'm happy to see citation of this work. And I also want to mention I'm very happy to see my former student Eric Wang, a scientist in Woodsboro now, a lot of excellent study in this area. Okay, let's move to this so-called ocean acidification mooring at the Grace Reef. That's actually right here somewhere. Yeah, at this 18-20 meter depth. So that's supported by NOAA's ocean acidification program, and this mooring is one of the 2 longer one in the east coast. We have now 8 years of data. So this one in the Grace Reef and another one in the right offshore University of New Hampshire by Joe Salisbury. And this is atmospheric CO2, this water CO2. Of course, you see this very strong seasonal control in the southeast following this SST, the temperature change. But interestingly is when we deseasonalize this data, there's a trend of rapid CO2 increase. Actually, this is very surprisingly high, about 10 microatmosphere per year. And if you know that the anthropogenic CO2 increase in the atmosphere is only about 2 ppm, so it should lead to about 2 microatmosphere increase per year. But we see from the current analysis of this data, we got 4 to 5 times higher CO2 increase, which means a much rapid pH decrease that you would expect to see from just responding to atmospheric CO2. And the reason right now our analysis attributes that to the wet-dry cycle of the river export in the southeast. It's this, we were in this dry cycle before 2007 and after 2008. We get this a lot of signal coming out of the CO2 from river and salt marshes, and I think that's the reason. And we just submitted this paper. My postdoc Janet Weimers submitted this paper to JGR Ocean, and we are now following this study and see how this changes in the dry wet cycle from the terrestrial export would affect us CO2 and pH signal, and likely it's possible, we'll get much less changes in the next, when we analyze next few years data. But one thing tells us this subdecadal changes, a lot of reasons could lead to that change. You can have much faster local response to ocean acidification because of various other reasons. All right, let me now move to this my study in the Gulf of Mexico, and I just want to use that as an example how this anthropogenic CO2 leads acidification can interact with eutrophication and respiration. So the Gulf of Mexico is a real good example because it's not just one system, and globally you have lots of places where have this double stressor, this anthropogenic CO2 and eutrophication. So this is a study my group did a survey in May and August 2007 that you can see when stratification starts in May, and when it comes to August, you have widespread low oxygen hypoxia events developed, and accompany that you have low pH and very low and very high DIC. And one reason we are sure that this very high DIC is related to very high production in the surface and the organic carbon decomposition lead to this is when you look at alkalinity, there's no alkalinity increase. So it's not like an advection or there's no alkalinity change. Actually, it's a little bit lower because accumulation of fresh water. So it's not like the west coast case, you have change in ocean upwelling or mixing in that case. So you can see aragonite saturation reduced greatly compared with May, but it's still relatively high. That is because the Gulf of Mexico system is actually a best buffered system with very high temperature and salinity, very high alkalinity. But we will see later when you superimpose this with continuous atmospheric CO2 increase when it comes to the end of 21st century. Actually, this entire area, if we still have the same eutrophication and hypoxia, this entire area will be undersaturated to aragonite. So that will have very serious consequence. So here I want to show you this correlation of pH to the oxygen consumption. So when oxygen decreases, you actually have this decrease in pH, and not only in the Mississippi and in the Changjiang, the Yangtze River plume area. So it's the same thing, and actually it follows theoretical prediction very well. When you have oxygen consumption, you will produce CO2. And the model would predict that pH change, and the data actually agrees with that in general. And now, if you only look at this picture, you probably will ask, where is the anthropogenic CO2? So that we will have to actually use a little bit of model tool to do that. Is we start with today's atmospheric CO2, and if we back to pre-industry, or we increase PCO2 in the atmosphere to the future, and then we could add this respiration-derived CO2 and drive the pH down. And that's what we showed in the previous slides. Now, what I want to emphasize is this is what we define as ocean acidification in open ocean or in the open Gulf Mexico. What here is we would see a 0.11 pH unit decrease by increased atmospheric CO2 from 280 to 385, okay, ppm. And but if we do this in this hypoxia zone where oxygen is really low or zero here, and where CO2 is already high, okay, and you could have, because of respiration, so you could have a much greater pH change due to this introducing this CO2 from atmosphere. CO2 increase in the atmosphere would give us an OA effect of 0.16 instead of 0.11. So that's quite a bit of increase. Actually, this will make this ocean acidification what we predict for the open ocean, the surface water, to the end of this century, actually already occurring here in the bottom water of Gulf of Mexico, and we can predict that for the future you will have a much larger ocean acidification signal than the oxic low CO2 water. And so that's what I call it enhanced ocean acidification because of this CO2 introduced from respiration, and you can call that as the other way, this ocean anthropogenic CO2 amplify the biological signal. It's the same thing when you discuss that theoretically. The reason behind that was this weakened buffer capacity in CO2. So if we start, I have this here is delta pH relative to a certain delta DIC change. So here is I multiply 100. So for every 100 micromole of DIC or CO2 addition to the system, how much is the pH change? So in the pre-industrial time, when or in today's oxic water with relatively low CO2, that change is relatively small, as we predict, like 0.1 pH unit or a little bit higher than that. And when we come to this anoxic water, and that with the same anthropogenic CO2, and you will have a lot stronger response. Okay, so for per 100 micromole of CO2 addition, we will have a lot more pH change. And the reason behind that is really the system has a lot weaker buffer capacity when you add the CO2 to that. And this is going to continue. This is another curve called the Revel factor. That's the oceanography like to use, but I just directly calculate delta pH against DIC. So this will continue, the signal will continue to amplify until a day when the water DIC, total dissolving organic carbon, surpass to equal or beyond this alkalinity value, and that will be quite a while later after the end of the century, and the signal actually will decrease in this, amplification will decrease, but the acidification is still there, of course. So overall, it's because of the weakening of buffer capacity, and I will discuss in more detail in the next part, is try to discuss how would we expect to see where under water salinity and temperature condition, you will see the largest amplification, that is the largest interaction between anthropogenic CO2 and metabolic CO2. That's the focus on my next part of the talk. I think we're a little short of time. I should try to go fast here. So I started with just an ocean water and river water mixing. Okay, just assume this river water has a low alkalinity and ocean water with high alkalinity, and then this aerobic respiration. But in real case, it will be more complicated. So in this work, Bill Sander and myself actually examined this case is when we presented this as the difference of the effect of this respiration caused the change. Okay, this is presented as a log unit of CO2 change and carbonated change in saturation state, and this is pH. So as you can see in the lower salinity, so salinity 4 to 36, and the lower salinity and lower temperature, the blue color is 1 degree C, yeah. And you see the pH change and the the CO2 change and the pH change, they are greatest. There's large change in both pH and PCO2 when the salinity and temperature is low. At a higher salinity and temperature, there are less change in relative CO2 and in the decrease of pH. So the estuarine water is very vulnerable to ocean acidification and to the interaction of respiration with ocean acidification. And carbonate, this main reason was really a decrease of the initial carbonate concentration. I'm going to skip some slides because we're running out of time, but I just want to point out what is really the big difference between low and high salinity. Really, it's part of main reason is because it's initial carbonate concentration is much lower, okay? And that is because of K1, K2 of changes with temperature and salinity, the K2 mainly. So that is a solubility, the dissociation constant of the carbonic acid, the K2, as a function of salinity and temperature. And that leads to a much weaker buffer capacity and lower carbonate concentration in the low salinity and low temperature water. And next slides really just try to say that these two systems, one at a low temperature and one at a high temperature, and how this will respond to the future, atmospheric CO2 increase. And it shows a system like Gulf of Mexico. It will continue to provide a high buffer capacity to neutralize the increased CO2 in the atmosphere, and a system in like a low pH, low temperature, like the Arctic and ocean, and like winter time, the Chesapeake Bay, at a very low pH. It has a very weak buffer capacity, and that system almost used up its buffer capacity. So it will change it in a different way from Gulf of Mexico. But still today, the system in this weak buffer capacity system has a stronger response, have a greater delta pH change due to the respiration. All right, I think I'm running out of time, and I'm going to just point out this key point that's all related to the initial carbonate concentration in the water. I'm going to skip this. This will just try to show that at this point that carbonate equal to CO2 concentration, the system is most sensitive to adding additional pH, so it has the largest pH change. I want to show one more slide that is a modification of this model. When we talk about estuarine in the discussion I just gave, is we assume at every salinity temperature the water is fully in contact with the atmosphere before we start this respiration. But in reality, that may not be the case. In an estuarine, we could have a system that either because of very high PCO2 in estuarine water, or this low PCO2 like in Chesapeake Bay, and high is like in Georgia River. There's no chance for CO2 directly embedded into the water. But even at a low PCO2, if the water residence time is so low, you may not have enough time to uptake anthropogenic CO2. So in this case, the ocean acidification signal, the anthropogenic CO2, is purely via the mixing of this already acidified source water, that's the seawater, mixing into ocean. In that case, you will have a zone in the estuarine that you will have a maximum pH decrease. Okay, so in the highest salinity area, we have this strong ocean acidification signal, but the water is well buffered. When you go in to mix with river water, buffer capacity decrease, so the signal delta pH change increase. But to a certain point, there is a value, this maximum response actually start to decrease. That's because we have less and less anthropogenic CO2 from the ocean gets mixed into the estuary. So there is this zone, weaks the buffer capacity. We can call that there is a maximum acidification zone exists. And this idea was advanced by Dr. Xinping, who currently an assistant professor at Texas A&M University Corpus Christi, and was presented in this paper. And the challenge, real challenge, is really how do we, prove that there exists this estuary maximum acidification zone. So that's an interesting topic we could follow. So I want to just quickly summarize. Respiration often plays a major important role in acidifying coastal bottom water today. However, anthropogenic CO2 uptake from the atmosphere will play an increasingly important role in acidifying coastal bottom water. I often hear people say, if respiration is dominating today, why you even study ocean acidification in the estuary? I disagree with that, as you can see from my talk. First is they have this interaction that will actually intensify the acidification. Secondly is because you have, the anthropogenic signal is going to keep increasing. So later this century, that actually they're going to surpass this respiration, and their interaction actually keep growing. And there is a strong enhancement of acidification in CO2 enriched water, and particularly at a low salinity and temperature, inside estuary in the low salinity water. Okay, I think I'm going to wrap up here, and as I understand, this talk will be uploaded, and you can see some other slides on my recent work, and I'm not going to present that anymore. Okay, thank you.
Wei-Jun, thank you very much. That was an excellent presentation. And what I would like to do at this point is open it up for questions. And we do have one question in already, and this is regarding your slide that showed the mooring time series plot off of Georgia, and the one that demonstrated the strong freshwater runoff in 2006. And it was noted that there was no corresponding changes in salinity or pH, and the question is why?
Actually, the large the increase in river discharge after 2008. So 2006 was still a dry year, and if we will go back to, since you can't see my screen anymore, right? Actually, there is a salinity. The salinity, there is no secular trend in salinity if you look all the salinity signal. But if you only look at the summer signal, actually there is a salinity decrease due to the increased freshwater output. And I didn't mention our rapid PCO2 increase was really summertime only. When you look at wintertime signal, there's no PCO2 increase. So that's another reason it's related to this freshwater export, and maybe also related to how close Gulf Stream is from the shore. Because wintertime, yeah, I think that's my answer. We are still continue to follow up that issue. Yeah. Thank you.
Okay. Are there any more questions? If you have them, you can type them into the chat, into the question box. Okay, I'm not seeing any more questions at this point. All right. Well, I would like to thank anybody or excuse me, thank everybody for attending the SOCAN State of the Science webinar today. If we welcome any feedback on this, and if you have any further questions or suggestions for topics on this webinar series, you can enter that information on the first web link that you see on the screen. And you can submit input by replying to the follow-up emails, and you'll receive emails from us listed on this slide there, Abby. And the recording of this webinar and a PDF of the presentation will be available on the SOCAN website with the URL listed also on this slide. So our next webinar will be Understanding Larval Bivalve Responses to Ocean Acidification, and that will be given by George Waldbusser from Oregon State University on Tuesday, April the 7th at 12 o'clock Eastern Time. And we hope that you can join us for that. And again, if you have any additional questions that come up for Wei-Jun, please send them to us, and we will make sure that he gets them and he can respond via email. Wei-Jun, thank you very much for a wonderful presentation today, and thank you all for joining us on our second SOCAN webinar. Bye-bye.
Thank you, Paula.
Thank you.